curve in itself should explain the allosteric nature, or that something is binding other than the substrate, or maybe the substrate also, that changes the activity of the enzyme. Okay? If we compare it here, okay, just keep adding substrate progressively, okay, and the activity of the enzyme keeps increasing till it plateaus out. Okay? But in this case, the activity, it gradually increases, okay, once the substrate concentration is increasing. Okay? And maybe there is a point wherein there is a particular substrate concentration which changes the conformation of the enzyme, or maybe it's from the very beginning that as you keep adding substrate, the conformation of the enzymes keep changing to progressively bind more and more of the substrate, and then you have a huge spike in the activity or the catalytic activity of the enzyme, and then obviously there is a certain substrate concentration at which all the active sites are bound, and then the activity plateaus out. Okay? So this is the point here, which basically gives us an insight into what an allosteric enzyme is. Okay? Now, keep in mind that the effector that we are talking about with respect to this allosteric enzyme can be the substrate itself. Okay? We'll be talking about how substrates affect the enzyme activity. Okay? So it can be the substrate. Once a single substrate molecule has been bound to the active site, okay, it changes all other available substrate binding sites on that enzyme so that it can bind more and more of the substrate and show an increased catalytic activity. Okay? Or it can be a positive effector. Okay? A positive effector is something that binds somewhere else on the enzyme and increases its activity. Or it can be an inhibitor, something that decreases the activity of the enzyme. And the examples are here. So if you look at the red line, which is the activity of ATCase in the presence of CTP. CTP is the final product of the enzyme pathway. Okay? And in a feedback inhibitory model, it inhibits the activity of ATCase. So if we look at as compared to just the normal control, if you have CTP along with the substrate, which is aspartate, you can see that the curve is further to the right. What does that suggest with respect to Vmax and the substrate concentration? Exactly. So if, say, 2 is the point which we randomly consider as the half Vmax of the enzyme. Okay? So if at, this is 5 notice. Okay. So if at 5 millimolar of aspartate, the enzyme ATCase achieves its half Vmax, if you have the same enzyme in the presence of its inhibitor, that is CTP, then it moves further to the left and we have something like 8 millimolar of aspartate which is needed for it to achieve half Vmax. Okay. Now, like I said, the effector can be a positive regulator also, something that increases the enzyme activity, which in this case, very interestingly, is ATP. Okay. And ATP, so if you look at the addition of ATP and the green line, you can see that the line has now shifted more towards the left. And with respect to the substrate concentration, if you still look at 2 as the half Vmax, something like 3 millimolar of aspartate will give you the same half Vmax. Okay? So that's the idea behind allosteric enzymes, that they can bind to various different effectors, inhibitors, and the activity changes due to the change in the overall conformation. Now, what makes ATCase a prime example of allosteric enzymes is the binding of both ATP and CTP. Okay? If you look at the structures of ATP and CTP, they are very similar. Okay? And both ATP and CTP bind to the same site. Okay? So if CTP is present, there is a certain conformational change which decreases the ability of the enzyme to bind to aspartate. Okay? If instead of CTP, ATP is present, it actually changes the conformation such that the enzyme has a higher affinity towards aspartate. Okay? Now, these look very similar to competitive or non-competitive inhibitory models, okay? But they are not. Okay? The idea that the enzyme does not straight away starts, increasing the production of CTP immediately. Okay? That there is a certain amount of delay, and there is only a certain amount of concentration at which the enzyme will change its conformation and then show a spike in its activity. Okay? The spike which is immediately seen is missing in case of allosteric enzymes. Yes?
So in this situation, ATP is an allosteric enzyme?
ATP is an allosteric enzyme?
Yeah, that's my question.
Does ATP catalyze anything by binding to it?
Yeah. Sure.
Does it?
Well, it seems that way.
Is ATP a protein that does that? Or is ATP something that is required for enzyme activity?
Is it just a requirement?
It's a requirement. Okay. Again, let's define what an enzyme is. What do you think is an enzyme?
I know what ATP is, it was the way it was phrased. I was wondering if it was now classified as...
No. Again, the enzymatic activity, that is the production of CTP ultimately. Okay? Or mainly, if we just talk about ATCase, that is carbamoyl aspartate. Okay? The production of that product can be done only by ATCase, not by ATP, not by CTP or anything else. Okay? So the enzyme, the protein molecule, is the one that shows that activity. How is that activity affected by different molecules that can also bind to that enzyme? Okay, is what we are looking at. So CTP is an inhibitor for ATCase. ATP is an activator for that same enzyme. Okay. Anything else? Any other confusions at this point? No? Okay. Now, because of the fact that they do not follow, the allosteric enzymes do not follow the Michaelis-Menten kinetics, we've classified them into two types, the K system enzymes and the V system enzymes. Okay? K to do with the kinetics, V to do with the velocity. So K system enzyme, the half Vmax, okay, changes in the presence of inhibitors or activators. Okay? And because they are not following the Michaelis-Menten kinetics, this half Vmax is not called Km, instead it's called K0.5. Okay? Keep this in mind. Wherever you see K0.5, you should get an immediate idea that we are talking about allosteric enzymes. Okay? And the V system enzymes, the inhibitors or activators, the binding of them changes the Vmax but has no effect on the K0.5. Okay? Very simple. It's just that these definitions are regularly used in terms of allosteric enzymes. That is why I just wanted to introduce these terms. What is a K system enzyme? What is a V system enzyme? Okay? And again, it's not half Vmax is the substrate concentration at which the enzyme has half maximal velocity, but it's not called Km in the case of allosteric enzymes. It's called K0.5. Yes?
So if the Vmax changes, that means that where our graph ends at the top is going to get higher or lower?
Okay. So the Vmax, again, like I said, any enzyme, okay, even if it slows down or even if it acts really fast, at a particular concentration, it will have a limited number of substrate binding sites. Okay? 1 molar of enzymes will have 1 molar substrate binding sites. 0.5 molar enzyme will have 0.5 molar binding sites. Okay? The idea is, even if it slows down in the presence of an inhibitor, or if it increases its activity in the presence of an activator, it will ultimately, given the time and the substrates, will reach its Vmax sooner or later. Okay? So you can see here at that point, every enzyme, once all the substrate binding sites are filled up by the substrate, every enzyme reaches its Vmax. Again, do not think of the Vmax in terms of the speed or the time. Okay? Think of it in terms of the substrate concentration. Okay? If you do that, it will be much easier to, grasp. And that is why, because every enzyme sooner or later reaches the Vmax, depending on the substrate concentration given the time. That is why we do not really use Vmax. Instead, we use half Vmax, which is a more, workable number. Okay. Clarifies your point?
Yeah, but Vmax still doesn't change, so 1/2 Vmax in my mind wouldn't change.
Why wouldn't it change?
Because
Go ahead. Look at this graph and, okay, like I said, consider 2 as the point which is half Vmax, okay, on this side. So, something like 3 millimolar of aspartate as the substrate concentration at which half Vmax is attained under controlled conditions. Okay? And now if you look at the presence of an inhibitor or the presence of an activator, do you think that the Vmax is changing? Half Vmax is changing? In terms of
concentration.
Exactly.
Yes. Okay.
Yes.
I was thinking in terms of the in terms of the x-axis.
There is a reason why these terms are defined in terms of substrate concentration. Okay. All right. Again, the definition of a K system enzyme and a V system enzyme is clear. Again, if you are talking about allosteric enzymes, do not use the term Km in your answers. It's K 0.5. Okay? Or K to the base of 0.5. Now, a few more definitions. So, any substance that changes the quaternary structure of an allosteric enzyme is called an allosteric effector. Okay? And for the binding activity or the change in the quaternary structure of the enzyme, an allosteric effector can be the substrate, it can be an inhibitor, or it can be an activator. Okay? Anything that changes the quaternary structure or its binding changes the quaternary structure of the enzyme is called an allosteric effector. Okay? Now there are 2 types of effectors. There are homotropic effectors and heterotropic effectors. Homotropic effectors, as the definition says, it's the binding of identical molecules, usually the binding of the substrate. Okay? The first molecule of the substrate, as soon as it binds the enzyme, or one subunit of the enzymes, again remember, many allosteric enzymes are a collection of different subunits, and each subunit has the enzymatic activity or the catalytic activity, has substrate binding sites. As soon as the first substrate molecule binds to the enzyme, okay, at one subunit, it changes the quaternary structure or the conformation of the enzyme, and it changes the active site, or it changes the ability of the enzyme to bind to the active site, the substrate to bind to the active site for all the different subunits. Okay? So, if 1 or 2 substrate binds, it changes for the whole enzyme, all the possible subunits, and that is an example of homotropic effectors. ADCase and the binding of aspartate to it is an example of homotropic effector molecule. Okay? Aspartate in the case of ADCase is an homotropic effector. And heterotropic effectors, if different molecules are binding at the same time, and then they are affecting the quaternary structure, okay, They are example of heterotrophic effectors, which in case of ATCase would be either ATP, CTP. Okay? If ATP is present, CTP is also present in the system. Aspartate is also present in the system. All of them are binding at the same time. Whichever is binding first changes the activity accordingly. Okay? And that is called heterotrophic effector system. Okay? Again, just pure simple, very simple definitions. Don't really have to go into too much of details. Again, just remember, in case of allosteric enzymes, anything that binds to the enzyme and changes its quaternary structure, changes its conformation state, is called an allosteric effector. And it can be either the substrate, either the inhibitor, or the activator. Now, this is important. Time. The two models of enzyme activity, or the models of enzyme activity with respect to mainly allosteric enzymes. Okay? Now, like I said, there are two, there can be many subunits to allosteric enzymes. Okay? Now, if we look at the enzyme as composed of 4 different subunits, each subunit having the catalytic activity. Okay? The concerted model, okay, it was it's by Jacques Monod, the same guy who gave the Lacoperon, okay, and a couple of others. Okay? What they define as 2 states of the enzyme. Okay? One is the T state, and the other state is the R state. The R state is the relaxed state, and the T state is the taut or the tight state. The relaxed state is the active state of the enzyme when it is able to perform its enzymatic activity. The T state is the conformation of the enzyme, or the 3-dimensional conformation of the enzyme, when it does not show, or it shows very low enzymatic activity. Okay? And the concerted model is very simple, very straightforward. Okay? What it says is that as soon as, so if the enzyme, it is existing in the T state, or the taut state, where it does not show any activity, as soon as there is the binding of a single subunit molecule to a single subunit, it changes the conformation of all the subunits in the enzyme. Okay? So once a single subunit binds to a single subunit binds to a substrate molecule, the conformation change takes place in all the possible subunits of that enzyme, and all the subunits are now able to bind to the substrate. Okay? So once a single substrate is bound, it changes the conformation of the whole enzyme. Okay? Otherwise, the other model, the sequential model, or the linear model, what it suggests is that it does not all happen at the same time. Okay? Again, these are based on observation by different scientists looking at different enzymes. Okay? And again, the way an allosteric enzyme behaves cannot be fully explained by either of the models. Okay? Some enzymes show a concerted model of activity. Other allosteric enzymes show or display a sequential model. In the sequential model, what it says that once a single subunit binds to a single substrate, it changes the conformation of only that subunit. Okay? And then you need another substrate molecule to bind to the other subunit. That changes its conformation. Then you need another substrate molecule binding to another subunit. That changes. And finally, you need the last substrate molecule to bind to the T state of the last subunit, and that changes into the R state of that enzyme. Okay? So it happens sequentially. All the subunits will not change their activity or will not change their conformation in one go, as in the concerted model. Okay? Now, there is the concerted model by Jacques Monod had the definition of T and R defined in other terms also. So it says that there is this term called L, which is equal to T by R. Okay, the amount of the substrate upon the amount of enzyme which is present in the relaxed state is defined as L. Okay, and how they came up with this model was that they were looking at a particular enzyme, and so okay, just going by this definition first, we'll come to their experiment later. L is equal to T by R. If L is very large, what does that suggest?
T is large.
There's not much going on.
There's not much going on, or the enzyme is present in its tight state where it is not showing a lot of activity. Okay, and if the L is very low, then there is more of a relaxed state, and the enzyme is showing very high activity. So you'll find this example online. You'll find their graph online, and what they basically did was with this particular enzyme. No, it was just a particular model. So if we have a graph like this, okay, what they found was that if this is the substrate concentration, this is the V. Okay, they found was that at presence of different effectors or the presence of different substrates, same enzyme behaving differently. Okay, and this is where they came up with this model, was with respect to substrate concentration only, was that as you keep increasing the, so here the L is, L is what do you call it, very low. Here L is extremely high. Okay, and this is how they basically discovered, or they decided to call the activity as a concerted model, was that based on this observation, and they came up with this L definition later, but what they found was that at different concentration of the substrate, okay, and in the presence of different effectors or inhibitors, the enzyme, an allosteric enzyme, can very much behave like a Michaelis-Menten or a non-allosteric enzyme, okay, or there are different concentrations of the substrate, inhibitors, effectors, where the Vmax will keep changing. Okay? And since they did not have, since they knew that the binding of the substrate, the effectors, the inhibitors and activators was affecting enzyme activity, so they said that there has to be two states of the enzyme wherein the quaternary structure changes. Okay? It is either the tau form, wherein it is showing very low activity, or it is the R form, where it shows very high activity, and the different graphs or different lines on the graph that they saw, depending on the different concentrations, they said that the enzyme has this new definition called L, which is the ratio of T upon R, and depending on how much the enzyme concentration is present either in the T form or in the R form, you'll see differences in the activity. Okay? And the sequential model people, they did not see these particular, gradations in their graphs, so they said that it's not happening all together, but it's happening one by one. Okay? Each subunit is affected by substrate binding in a linear fashion, one after the other. Okay? The concerted model and the linear models make sense, or the sequential models?